Hello everyone, Jeff the Green Review here. Some chemicals plants produce are similar in function to hormones in animals. Sometimes called phytohormones, they have control over functions of plant growth and are called plant growth regulators. These chemicals are produced in extremely low concentrations. They're not produced by specific organs or glands the way animals produce hormones. They're produced by many cells throughout the plant. Because of the low concentration and diffuse nature, phytohormones have been difficult to study, and more are being discovered. There are 6 general phytohormone groups, with the last one being a catch-all group for recently discovered phytohormones. Some of the groups include many different chemicals found in different kinds of plants, but they're grouped together based on their effects on the plants. Each of the hormone groups has positive and negative functions and often react and counteract with another group. The first group is the abscisic acid or ABA group. ABA is produced in the leaves and it acts to prevent buds and seeds from growing at the wrong time of year. It accumulates in seeds as the fruit matures, preventing sprouting in the fruit. As its concentration lowers through washing away with water and breaking down in cold temperatures, buds and seeds are released from dormancy. ABA is part of the chemical reaction that closes stomata when a plant is under stress from lack of water. Auxins are chemicals produced in the apical meristem tissue at the tip of a branch. High concentrations move down the stem to prevent the other buds on the stem from growing. Pruning the tip of a branch reduces the amount of auxin in the branch, releases the other buds from chemical dormancy. Auxins produced in seeds cause the plant to develop a fruit structure around the seed. They also control the growth of stems, roots, and flowers. Auxins are produced more on the shady side of the stem, causing those cells to grow faster and forcing the stem to bend towards light. Cytokinins produced in plant roots influence cell division, internodal branch growth, and leaf growth in the top of the plant. They also help keep the root system and the top system of a plant in balance. Removing roots reduces top growth. Cytokinins work together with auxins for controlling growth, and they counteract apical dominance. They work together with ethylene to cause flowers, fruits, and leaves to fall off. Other organisms like bacteria, fungi, insects, nematodes, and viruses can produce cytokinin chemicals that spur the growth of plant cells. The plant then produces a growth such as a burl, a gall, or witch's broom that may damage the plant. Symbiotic relationships between plant roots and nitrogen-fixing bacteria or mycorrhizal fungi are created with cytokinins. Ethylene is a gas found in all plant cells, but also, being a gas, it's continually escaping the plant. It affects cell growth and the cell shape, such as preventing leaves from expanding until they have enough light. When tree trunk cells flex in the wind, ethylene production causes thicker cell walls. Ethylene gas builds up in fruit, causing the fruit to ripen and release the seeds. Ethylene gas from a ripe apple or banana in a bag with green tomatoes will cause the tomatoes to ripen faster. Wholesale storage units that keep fruit for months at a time need to continuously remove ethylene gas, or else the fruit would finish ripening and begin to rot. Gibberellins are chemicals that regulate enzyme production that in turn break down stored food reserves within the seed's endosperm, which are utilized by the growing seedling. They also promote flowering, cellular division, and reverse the inhibition of shoot growth and dormancy caused by ABA. Gibberellins help control internode length. Low levels of light cause the gibberellins to increase internode length, making the stems grow longer. Low levels of gibberellic acid or gibberellins cause the plant to be smaller. Florist flowers and some landscape plants are sprayed with chemicals that suppress the gibberellin production, so they stay small and may not need pruning. Gibberellins also are used to increase the size of seedless grapes. Among the last group of hormones are the newly discovered ones, such as salicylic acid, that activates genes that produce chemicals that help defend against pathogens. Also, some of the new ones have been discovered while researchers were looking for anti-cancer drugs, such as sodium salicylate, that has been found to suppress several different kinds of cancer cells. There are many other plant hormones, and new ones are still being discovered. So what's this mean to me? Ethylene gas escaping from fruit, especially apples, bananas, and pears, will cause nearby plants to react. Placing an apple in a bag with green tomatoes helps them ripen faster. Placing a fruit bowl near a vase of roses will cause the flowers to die much faster. Fruit stored in warehouses and even trucks during shipping can release so much ethylene gas that it causes all of them to decay. These facilities have vent fans to prevent this from happening. In other words, what does this mean to me? Auxins preventing growth of other buds is called apical dominance, and it causes plants to grow more upright or columnar. Tall, skinny trees may not produce much shade, but as a street tree, they're less likely to cause damage to buses and trucks. So we do want apical dominance on our street trees a lot of times. Apical dominance also prevents the buds along a horizontal branch from growing. If an animal eats the tip of the branch, it does not stop the branch from growing, thus reducing its food supply. It does the opposite. It releases the bud to grow, thus producing many new side branches that are potential food for the animal. If the tip of the branch is pruned away, then there will be more side branch growth that could cause more work for the pruner. Knowing that side branches are about to be released will cause the prudent pruner to calculate where the pruning cut should be so as to direct the side branches toward a direction that causes less work and is a benefit to the plant. And another, what's this mean to me? Because auxins are in charge of stem and root growth, they have been synthesized for various uses. They are toxic to dicot plants in high concentrations because they cause the plant to grow faster than it has the resources to support. They are less toxic to monocots, so weed killers using auxins like 2,4-D control weeds in grassy situations like lawns and cornfields. On the other hand, auxins like whatever this says, to NAA and indole 3-butyric acid, BA, are used by nurseries and homeowners to stimulate root growth when taking cuttings of plants. Gardeners can have more success when propagating plants from cuttings taken from leaves, stems, or roots if auxins are applied as a rooting compound to the cut surface, where they promote the root initiation. Auxin promotes tissue formation in grafts, which joins the surfaces of the graft together. In tissue culture propagation, plant growth regulators produce callus growth and multiplication of cells and rooting. So there's a lot of man-made uses for these phytochemicals that the plant produces. And this is Jeff with the Green Review. Thanks for watching.